All right, hello everyone, welcome. We can go ahead and get started. So my name is Eliza. I'm the program manager at the 3Rs Collaborative, and welcome to this little webinar series. I'm just going to give a quick overview of our organization and this series to start. And I'll start with a disclaimer that I'll read right off the slide. This webinar and workshop series is made up of companies who may be competitors or customers of one another. Accordingly, nothing discussed during these webinars and workshops are intended to result in an agreement on price, exclude suppliers from any market, or otherwise restrain competition. Those participating in this webinar and workshop series are instructed to avoid discussion of competitively sensitive subjects, including costs, prices, sales, product marketing, and other confidential information. In addition, neither the 3RC nor the IQ will use contact information received during this collaboration for marketing purposes or engage in marketing or sales conduct during collaboration activities. And now we'll just give an introduction to the 3RC and the webinar series. So the 3Rs Collaborative MPS initiative aims to accelerate the adoption of MPS in scientific research. And we do this by, we seek to increase adoption and regulatory acceptance of MPS in coordination with animal studies. And this is just our board of directors, and our members span across the scientific field from industry, academia, and government agencies. And the MPS initiative specifically includes over 50 member institutions and over 75 members. And we aim to provide thought leadership and expert consensus from developers combined with other stakeholders on implementation of MPS, facilitate appropriate discussion, collaboration, and sharing between developers of MPS, create direct engagement between developers and regulatory agencies on the use of MPS, develop external partnerships and collaborations to provide perspectives from developers throughout the field, and create resources to facilitate increased engagement with developers. And we accomplish these goals via 3 work streams. We have a regulatory work stream, technology expo, and end user and education. And I'll just go over briefly what our work streams have been doing. We've been engaging consistently with end users and providing education. In 2025, we've been hosting quarterly workshops where we talked about using AI and machine learning in MPS, retina ocular MPS, multi-organ MPS, and now today's overview of the ISTAND program. And we have one more webinar in December. We've had posters at conferences and continuer submissions to All Text Corners, and we continue to organize additional webinars on relevant topics. And you can see our past workshops and webinars on our presentation page on our website. And I'll go over our regulatory group progress. In 2025, they published a perspectives on using MPS for regulatory applications, and the QR code for that paper is on the screen. They've promoted at various conferences. They are continuing forward progress on a cross-platform DILI project and beginning the development of the benchmarking project for MPS end users. In 2021, we launched the MPS Technology Hub, and we continue to add disease areas and tissue filters and continue to regularly promote it. And this summer, we launched a new publication hub where you can explore relevant publications by our member companies, and you can filter by tissues or disease applications. And we'll be continuing to add more publications to that. We have one more workshop for the year, which will be at the beginning of December, so stay tuned for that. And during this webinar, you can use the Q&A function to ask questions, and we will be answering those questions at the end of the webinar. And you can visit our website to learn any more, and my email is on the screen. If you're interested in joining our MPS initiative or becoming one of our sponsors, just feel free to shoot me an email. And now I will go ahead and stop sharing my screen and bring on David from the IQ.
Thank you, Elisa.
Yeah.
I'll go ahead and share. Hopefully that looks good. Okay. My name is David Kukla, and before we get everything started, as Elisa said, I'm just going to give a quick overview of the IQ MPS affiliate. So the IQ is a collaboration of pharma and biotech companies created as an affiliate within the International Consortium for Innovation and Quality in Pharmaceutical Development. And the IQ has four main aims. One, to develop external partnerships and collaborations to help enhance the inclusion of industry priorities, to serve as a thought leader for MPS developers and stakeholder organizations, also to provide a venue for appropriate cross-pharma collaboration and data sharing, and finally, to create a focused engagement between industry and regulatory agencies on the current status and the evolving field of MPS. So the IQ is currently composed of 26 member companies with over 150 active participants, which represent experts from drug safety, 3Rs, ADME, and PKPD. And on this slide, I'm just showing our 2025 Executive Committee. And then here is a breakdown of how we structure the IQ MPS. There's an executive committee, which provides leadership and guidance in the management of IQ MPS business and affairs, and implements strategic plans recommended by our steering committee. And the steering committee is made up of two representatives from each member company, and they are the primary decision-making body for the IQ. And then within the IQ, we have five major work streams, which I'll quickly highlight on the next few slides. But within each work stream, we also have several sub-teams which address specific issues. So the first work stream is regulatory engagement, and their mission is to build regulatory confidence in MPS and align on a common understanding of how MPS data should be handled in a regulatory submissions by sharing industry knowledge of MPS utilization with regulators and collaborating to overcome challenges utilizing MPS for drug development applications. The second work stream focuses on publications, and their mission is to, their mission is to outline the contemporary pharmaceutical industry perspective and considerations for developing, evaluating, and characterizing MPS models. And the workstream already has published on several organ systems and applications. Here I have listed several of the PubMed ID numbers, and there's several more to come. Then the pilot project workstream works to initiate near-term proof of concept studies for data sharing or prospective collaboration for qualifying MPS systems based on a specific context of use. And then finally, the strategic partnership and communications workstream aims to develop external partnerships and collaborations to help enhance inclusion of industry priorities by making IQMPS the partner of choice for pharmaceutical MPS education, alignment, and engagement. And if you have any questions, please feel free to email us at the email address listed on the slide, or you can keep up to date with IQMPS initiatives either by looking at our website or following us on our LinkedIn page. And with that, I'll turn it back to Elisa. Thank you.
Thanks, David. And we can go ahead and bring up our speaker, Dr. Tomita, if you wouldn't mind sharing your screen.
First of all, do you hear me?
Yes.
Okay,
Yes.
great. Let me bring up my screen.
Great.
All right. Thank you very much for inviting me to this webinar today. I was asked to introduce ISTAND program, accepting MPS and other technologies to this program. But I'd like to broaden my scope by a little bit and talk a bit more about how we would accept new technologies, new methods, more in general into regulatory submissions. This is a typical disclaimer that this view is my personal view, and it does not necessarily represent official FDA position. And as you can imagine, many of the applications are program specific. And before concluding anything from today's discussion, you may want to reach out to each programs or review divisions for their point of view. Starting this year, we reported or published three major reports from FDA and one from GAO. And first one, I'm sure that you are all very familiar with this report to reduce animal testing roadmap. And this had a quite impact on ISTAND program, as many of those methods would need new technologies or tools to reduce animals, and many of those would fit into ISTAND program. Second one is a GAO report. You may know that the GAO is a government accountability office commissioned by the Congress. And a few years ago, Congress was interested in MPS technologies and initiated the investigation of how policies, budget, and planning are made for MPS. And recently, this spring, they made a publication about MPS technologies, including achievements and some challenges regarding to MPS technology. And of course, one of the challenge was included as that regulatory acceptance of MPS is difficult, and I would like to talk more about those in the next few slides. And then lastly, this summer, we decided to convert our ISTAND pilot program to full program due to high demand and also quite strong interest from developers and industries. From outside, this change may not be much, but internally we have received more resources, and we will try to follow the PQP biomarker qualification program timeline. Typically, three-stage process would take 3 months, 6 months, 9 months, or 10 months timeline for the review. And we'll try to adhere to this timeline also for the ISTAND program. So for the consideration for the NAMs, today's discussion, I would more or less interchangeably use the NAMs and MPS because most of these concepts would follow very similar discussions. And we are strongly interested in NAMs to fill the needs to reduce animals and create new tools for the ability to predict safety risks in humans, and in some cases may be useful also for the efficacy assessments. And of course, CDER is committed to principle of the 3 R's, testing, animal testing, and I would think that ISTAND program and other tools that you are interested in will fill that gap. So earlier on in ISTAND program, we had very strong interest from tool developers, and they proposed many different types of tools. And many of them we had to reject because these tools are more focused on internal decision making by sponsors or industries. That is quite understandable because when you develop new tools, you do not have many experience and you don't have strong evidence, and you would like to use those tools in more earlier phase of drug discovery phase, and you would like to gain more experience. While FDA is more interested in later phase, primarily drug development phase, late preclinical stage to clinical phase, and something that FDA can make regulatory decisions on. So there was a discrepancy, but over years, I think it's getting better. And we have seen many good context abuse and drug development needs. So when you are formulating new application to ISTAND qualification program, or more in general FDA submission, please think about the drug development needs that is pertinent to regulatory decision making. And also context use should be reflective of those drug development needs or gaps. And so in this context, NAMs are used in regulatory submissions that will be slightly or quite a bit different from research tools. And when you create tools for regulatory purpose or regulatory decision making, they need to be very robust and reliable. So when you submit applications, we will expect that those tools are very reproducible, rigorous, and has applicability in the context of use. Before diving deep into the ISTAND program, I would like to describe there are many other ways to approach FDA besides qualification program. Of course, qualification programs include the biomarker qualification and clinical outcome assessment, ISTAND, and MDT, that stands for the medical device tool development. And also we offer pre-LOI and letter of support, which can take much shorter time than going through the qualification programs. And if you are interested, I'll be happy to discuss more about pre-LOI and LOS stage. So besides these qualification programs, you can also submit your data generated from NAMs directly into the IND applications. You can have a discussion with FDA with the type B, C, or D meetings. And we also offer pre-IND meetings. These are quite appropriate if these are new tools and FDA reviewers are not familiar with these tools, then you may want to bring in as a pre-IND meetings and get the sense of how FDA would review or accept those information. Another thing that I wanted to mention is the DMF or drug master file. Some of you might be tool developer and you'd like to share your evidence or data with the FDA, but you don't want necessarily to share those proprietary information with the users. In that case, you can use the DMF pathway, and where you can submit your evidence or data to FDA directly, and sponsors can refer to those data without disclosing them to the sponsors themselves. And of course, the benefit is that same DMF files can be used by many different sponsors or many different programs, so you don't have to resubmit the same data again and again. And of course, consortium like 3Rs and many others like critical path are a very important part of reviewing or accepting these new tools. And we also, within FDA, we also have other doors like CPIM and MIDT and CIDs. These are the non-qualification programs, but provides you the opportunity to interact with the FDA reviewers. And these are mostly one-on meetings compared to qualification that is more extensive, and you can have multiple meetings in a qualification program. I can pause here for 1 second if you have any questions, or I can just keep going. Right, if there's no question, I can just keep going. And
I think there's a couple questions that we could take. We can unmute microphones. Help with that. Cool. Yeah, just raise your hand. So Jay, go ahead. Do you have a I tried to unmute you.
Let's
try one more. Oh, it is really not working.
I think that's the user side.
Can you hear me now?
Yeah. Oh, yes.
Okay, yes. So how do I find out information and who do I contact about requesting meetings on NAMs for a pre-IND or a B, C, or D meeting? Specifically, somebody who knows something about NAMs can help. Is there anybody there who can help guide the process, can help, arrange these meetings? And what is the difference between a B, C, and D meeting?
B, C, D meetings are depending on the phase of the application. So, for example, type D meetings is more narrowly focused topic, while type C meeting is more broader, and type B meeting is for the end of the phase, like phase 1 or phase 2 or 3 of the clinical trials. In terms of pre-IND meeting, these are just go through the regulatory submission channel. So you will reach out to the appropriate clinical division to arrange those IND. If your tool is super broad and not sure which division you can reach out to, you can always reach out to us, and we can try to figure it out. There might be some kind of inbox for more broader questions, but if not, you can submit it to ISTAND box, and we can try to figure it out. And within FDA, not only the ISTAND program, we also have NAMS coordinating committee. So if you clearly indicate that your tools will fit into NAMS, so MPS, and then it's likely that when you request pre-IND meeting, those requests will go through NAMS coordinating committee instead of just focus on the single division.
All right, that was very helpful. Thank you.
Welcome. Rod, you have something that you can suggest?
Let me allow his mic. You should be able to unmute now, Rod.
Yes, thank you. So, and I just wanted to add on as part of that CDER NAMS coordinating committee, we are trying to leverage the critical path innovation meetings for early contact with either developers or sponsors that are interested in getting some input from CDER on their alternative method approaches. And I think we're also trying to develop something more formal to engage even earlier in the preclinical or in the non-clinical space. So that maybe we could provide a little bit more back and forth before the pre-IND meetings, when there are plenty of non-clinical studies still to be done, and unfortunately, a lot of our meetings in this space only occur after sponsors already done most of or all of that work. So it'd be nice to have some earlier interactions, and I think we're trying to figure out a path to do that now. So thanks.
Yeah, thanks, Rod. The other question that's come in via the chat from Ksenia is, where does fit for purpose initiative fit within slide 6? And then was linked to this as a DDT fit for purpose initiative. Don't know your.
Yeah, I'm not familiar with fit for purpose program So maybe I can ask somebody within FDA and provide information to this group later.
Great. Awesome. Thanks. Those seem to be all the questions for now. So I'd say go ahead and continue. Thank you so much.
Okay. So for the next several slides, I can show you the progress or current status of ISTAND program. So far, since the initiation of ISTAND program, we have received over 70 applications, including pre-LOI and LOI and QP submissions. And more than half of them are NAMS related, and that include also AI and DHTs. And then currently we have accepted 9 LOIs and 2 QPs, and those acceptance are issued as letter of determinations, and they are all posted in the link that's showing in the bottom of this slide. So if you are interested in any of these applications, you'll see their submissions, like LOI submissions and the QP executive summaries, together with how FDA reviewed and commented on. So these are included in letter of determinations. They are all posted and archived in this website. So I would recommend you to go there to check. Something might be similar to what you have, and that will be very helpful to formulate your context views or what kind of data we would request to have. So this is a diagram showing the current status, and as you can see, about a third of them are MPS related, and about a quarter of them are AI related, and there are many other types of NAMs in the mix, and we have DHTs and others. And this table shows all 9 accepted applications. 2 of them are QP. One is from Integral Molecular, and the second one is from Emulate. And among those, Emulate and LAMPs are MPS applications. And I would go more deep into the Emulate and LAMPs applications in a later slide. So I just wanted to highlight a few applications in this table. One is Integral Molecular, and which is very similar to Charles River Laboratories' heterogenics platform. These are used to assess the tissue cross-reactivity for antibody-based therapies. And typically, these are done in the tissues from various organs. And these 2 companies made the array, membrane proteome array in vitro system, and they can do the off-target binding of antibodies. And this technology has been progressed quite well, and in some therapeutic areas like oncology, they have been accepting these method as a replacement for traditional histopathology-based tissue cross-reactivity. Or maybe I misspoke. QP is a qualification plan, and the third stage is the qualification package. That's the last stage. So 2 QPs, a qualification plan is from Integral Molecular and Emulate. Another one that is the replacement for current method is the Fresenius Kabi. They have developed panel of in vitro assays to test the reachable for epidural administered products, and these reachable are coming from the product packaging. And typically, these are done in the rat studies, but they created the panel of in vitro testing that could replace these rat in vivo studies. I just wanted to show you the possible impact of FDA roadmap. So since 2020, we have received many applications over years, and we published FDA roadmap in the second quarter of this year. And as you can see that the application to ISTAND spiked right after the FDA roadmap. So one of the strong benefit of applying for codification is that it will provide you the platform to interact with FDA reviewers, and your application will be thoroughly reviewed by many different disciplines, including clinical divisions, farm talks, statistic analysis, and in many cases, CDRH. They are involved when the tools are related to in vitro diagnostic tool. Could be other medical devices like MRI and CT or histopathology. And then we have clinical pharmacology reviewers, and in some cases, CBER reviewers are also involved. And as you may know, that CDER and has research divisions, and they might be very familiar with your technologies. And if we have expertise in certain research tools, then we can bring in those experts as well. And these experts will form the what we call qualification review team. They meet regularly and create the recommendation for the drug development tool committee, and they are the decisional body to accept or reject the applications. So many applicants, requesters struggle with creating acceptable context of use. And context of use should be very concise description of a biomarker, or in this case, tool, and how they're going to be used in the drug development, and has to include two types, MPS or in silico, and it has to describe intended use in drug development based on the drug development needs. So first you have to define what is the current paradigm for drug development need, and what is missing or what is defective in that situation, and how your tool can supplement that gap or replace that assay. And often case, context of use would include patient populations, disease types, and models, and the stage of drug development, and the mechanism of action of the therapeutic intervention. Many requesters tend to create context of use that is very broad. That is understandable because you want to use your tool for many different purposes. But that would end up with a very difficult situation of reviewing those applications to validate your tool. And we would ask evidence or data for many different situations. So when you create context of use, you should be focused, and you should be thinking about what kind of evidence, what kind of data you can bring in later on in the qualification stage, so that you can validate your tool. And as I mentioned earlier, NAMs can be used for many different phases of drug development or drug discovery. And these tools can be used for discovery and preclinical and maybe early clinical phase, while FDA is more involved when these data are submitted to IND or NDA. So preclinical phase, later preclinical phase, and clinical phase are the purview for FDA. It's perfectly fine and perfectly reasonable that these tools have strengths in early phase, like discovery phase, because of the you can add more information, but these are not FDA purview, so you don't have to get FDA clearance or FDA involvement, and you can work directly with users, mainly pharmaceutical companies, to develop those tools. I wanted to provide two successful examples of how they formulated context of use. The first one is quite known by now, that emulate application. And we had quite a number of back and forth between emulate and FDA to come up with this context of use. And it narrows down the context quite a bit so that we can have reasonable expectations of how this tool can be used during development. So as you can see from highlighted words, it says greater evidence. That means these are weight of evidence for supplemental data. It's not the replacement of battery of POC studies that we request. So that will make it easier for the acceptance. If you want to create some kind of replacement for current battery of testing, that will be more stringent. And then it also says that these are used for enabling phase 1. So this is late preclinical phase, tox study. And one of the major thing about this human liver chip approach is that they will be used when there is a comparator, meaning that there is a compound or drug that displayed the DD in the clinic in the past. And these are called the comparator. And that therefore, the new related drug candidate has a priori concerns of DD. And what it does here is that when you run these comparator through the system, MPS system, they should be able to find the DD signal. If not, this MPS platform would not be able to catch those DD signal. So that is the safeguard of this context of use, because you can narrow the scope by using the comparator, and that can identify the positive signal when there is a known drug that displays DD. And then at the same time, that will also create a scale that when you see certain amount of signal, you can say that that is likely to cause DD. And if your new drug candidate is far below those DD signal, then you can assume that the new drug can be safer than the comparator. Second CO example is from PIT trace. This is NCAT's supported activity, and it's a liver SNES microphysiological system. And this is used in a clinical phase, clinical trial, when the you would need to include patients with a MASLD or some kind of hepatic impairment. And first, you run the trial without any liver hepatic impairment, and then establish the dose. But when you need to include patients with a hepatic impairment, you need to have studies done in between. And this MPS platform is designed to bridge those gaps by creating the environment you can induce masked situation on the chip, and then you can study how those disease would impact the hepatic clearance. And accordingly, you can reduce the dosing when there is a signal detected in this platform. So just to summarize, CDER recognized the need and the utility of NAMs, and we believe that it has capability to predict efficacy and safety risks in the humans. And context views for NAMs needs to be for regulatory decision making, and you can have NAMs for prior to this phase, but these are mostly done within the industry or users, and you do not have to come to FDA for those. And you can submit NAMs generated data directly in the part of IND, NDA, BLA submissions, or you can come to those technologies with the DDT qualification programs such as ISTAND. Lastly, we will protect your proprietary information when you submit your data. Of course, we will ask to submit the data and sometimes raw data together with the algorithms or code. And these would stay within the vault of FDA and we would not need to share those in the public. And that is the conclusion of my presentation. And I am listing the ISTAND email address at the bottom. If you have any questions, please shoot an email to this email address.
Thank you, Dr. Tomita. So we'll go ahead and take any questions. You can put them in the Q&A, the chat, or just raise your hand and I can unmute you, whatever you prefer. Okay. So Will, you should be able to unmute.
We don't hear you right now, Will, so you may need to try once more to unmute.
All right, how about that?
Yep.
Sorry about that. Hi, Will Proctor, GSK. Really nice presentation. I appreciate the background on the ISTAND. Just a general question around the really nice C of U for emulate and hepatotoxicity risk assessment. How, just thinking about this from a clinical application, how would this change an approach that a sponsor would take, if they're submitting this for first in human with comparative data? There's a little bit of background here. With comparative data saying we will have a reduced risk for, say, hepatotoxicity based on probably the totality of the non-clinical package, and then eventually to clinical data. Is that really, what will that do to help sponsors, work through hepatotoxicity risk? Would it change potential clinical practice? Would it change, how we think about causality in relation to, LFT elevations? I mean, it's a big question. I'm not expecting that on this call, but I think for some of us that are in the drug-induced liver injury space, we want, increased tools, but showing differentiation for a known competitor compound is something that we've been doing with simpler tools for a decade maybe or more, to justify why we would progress into clinical trials and to use our normal non-clinical approaches. I'm just curious what this qualification would mean for someone if they had that data. What wouldn't they have to do that they would normally do anyway under just the standard non-clinical approaches in clinical practice?
So this is a weight of evidence approach that I don't think it will replace any currently done testing or monitoring, but I think it meant to add some kind of comfort or assurance that something that found in the clinic before and replicated in this packet, in this platform, and then you don't see that anymore in the new compound. So it will add evidence that is safer than previous version of the drug.
Thank you.
All right. So next question is Ming, and I'll allow your mic. You should be able to unmute now.
Can you hear me?
Yep.
Okay. Hi, I'm Mingyi Huang from Arakari Biosciences. Very, very good talk. Just a question on the earlier slide where you emphasize has to be within the confines of FDA's purview, such as preclinical stage. In there, you had listed liability and PK/PD. Can you just clarify what you meant by liability in that context?
So I think the PK is I had shown example in the lamps example when you need a PK adjustment, and you can provide the bridge study by using MPS rather than you try to create dedicated clinical study.
And liability, that's part of PK/PD because you have liability in one and then PK/PD in second.
Sorry, liability can be a bit earlier stage, like I think that maybe Emulate is the good example for not related to PK/PD, but liability for toxicity that might be expected or concerned, and then you can reduce those liabilities.
Okay, you mean like an adverse effect?
Yes.
Okay, thank you.
I just wanted to add that at least many of those technologies, they claim that they can do much deeper dive in the cause of these adverse events or toxicity pathways. So compared to the other platforms, they might be able to provide more insight into why certain things are happening.
Yes. Yeah, that's one of the advantages of human-based MPS. Yeah. Thank you.
All right. Are there any other questions? Okay. Chris, you can go ahead and unmute.
Yeah, hi, this is Chris Inkley from Biogen. Thank you for the information. I wanted to ask a little bit about assays that would be used more in the discovery phase. Oftentimes these are efficacy models, things centered around primary pharmacology. Those do make it into regulatory submissions, but sort of, is that out of scope for ISTAND qualification? Or can you define context of use for assays like that? Maybe a set of rare genetic diseases impacting a certain cell type, and then put that forward for ISTAND.
Yes, there are many cases that those findings or results are important in a later stage, and I think that they are impactful and we are interested. I was talking more on some of the, let's say, MPS data that may be included in IND package, but they may not have very strong impact or weight on making regulatory decisions. And those are not, those are outside of purview for qualification program.
Okay, it looks like we have a question in the chat. FDA roadmap explicitly refers to naked antibodies. Do you see any NAM developments in the ISTAND program specific for this modality?
I mentioned one applications or two applications from integral molecular and also Charles River, and these are for tissue cross reactivity assays for the antibodies, and these technologies can be used for anything from monoclonal antibody all the way to cell-based like CAR T-cell type of applications.
Great, thanks. Any other questions? What is the process for requesting a pre-LOI meeting?
It's quite simple. Just send the interest email to the email address listed in my slide. I stand at fda@hhs.gov, and we will ask you to prepare a few slides describing what is your drug development needs and possible context of use. And if you have any preliminary data, provide summary of the data. I forgot to mention, but context of use is difficult to formulate, and if you uncertain, but you are sure that context of use would fit within the drug development needs that FDA is interested in, then you can just propose, and then we can work with you to have better context of use throughout the journey of qualification review. In many cases, context of use evolve from LOI to QP to FQP.
Any more questions? Okay. So we can go ahead and close out. Thank you all so much for joining, and thank you, Dr. Tomita, for speaking. I hope everyone thought this was very helpful. Thank you, everyone, for your questions. And yeah, everyone have a good day. Thank you.
Thank you. Bye-bye.
Bye.